Okay, today we're going to talk about the C6 unit, and that is going to be on photosynthesis and respiration. So, when we're talking about photosynthesis and respiration, there are going to be two key players into this, okay? Two cell organelles that you should be familiar with already. Number one, we're going to have this little guy right here. Called a chloroplast. And then on the other side, we are going to have another organelle called a mitochondrion. Okay? Now, inside of the chloroplast is going to absorb light and be able to do a process called photosynthesis. Okay? And photosynthesis, you are going to take a couple of different things. Number one, you're going to have carbon dioxide, and you're also going to need water. Okay? And so what is going to happen there is that that's going to go together, and then you're going to have an arrow that goes to the products. And the products of photosynthesis are going to be two different things. There is going to be glucose, or C6H12O6, and oxygen. And those two things are what is going to we're going to get from that chloroplast when photosynthesis is all finished. Now, on the other side with the mitochondria, what we're going to have is we are going to have what we call respiration. Okay? And respiration is going to use the sugar, and it's also going to use oxygen. And then the products of respiration are going to be carbon dioxide and water. Okay, now this might seem like, okay, what's really the purpose of memorizing all of this? However, it is absolutely crucial, and one of the biggest things that we want you guys to learn is we want you to know exactly what's going in and exactly what is going out at any given time during photosynthesis and respiration. So if you do not know the reactants and the products really well, you are not going to be able to do well on these tests, because most of the questions are going to have to do with what goes in and what comes out during different processes and stuff during photosynthesis and respiration. Now, the other thing that you need to be able to understand with this formula is that if you look at it, photosynthesis right here is the exact opposite of respiration. So really, if you just memorized one of these, you could do just fine with just knowing that the other one is just the exact opposite. So you can just switch the direction of the arrow if you really wanted to. Okay, so if you can see, these things are a cycle. And so when we're talking about a chloroplast and a mitochondrion, that they just work in a cycle back and forth between each other. Where the chloroplast here, this guy is going to be providing the sugar and the oxygen for the mitochondria, which will break that down, create energy for us, and then will put out CO2 and H2O that this chloroplast can then use to be able to make more stuff over and over again. So that is very, very important. Okay, in the second part, what we're going to be talking about here is that we're going to go into photosynthesis and respiration by themselves and kind of do some problem solving with that. So number one, in photosynthesis, we're going to have two different steps that happen during photosynthesis. The number one is called the light dependent reaction. Okay, and just like it says, it is going to be dependent on light. Really not that difficult when you look at that, okay? So, number one, what's going to happen is that you are going to have molecules inside of things called, well, let's get it ourselves a good, okay, so just give us a chloroplast to work with here. Okay, so number one, what we're going to start with is we are going to have the sunshine. Here, let's make that a little better. Little beautiful sun. All right, now that beautiful sun is going to come in and it is going to shine into this, and it is going to shine onto these little things that look green, and they are going to be called the thylakoids. Okay, and so the first part of photosynthesis, the light-dependent reaction, is going to happen inside of the thylakoids. Now, inside of the thylakoids, there are going to be pigment molecules that are going to pick up this light. And then, so here we have one of those, okay, and there is going to be an H2O, and there's actually going to be two of those that we start out with. And what happens is that, again, our light shines onto that, hits it, and it starts to shake as the light's hitting it. It starts doing the truffle shuffle. And as it's shaking, what's going to happen is that it is going to break apart our water, okay? And it's going to steal an electron. So it's going to get an electron. And then what you're going to end up with is you are going to end up with some hydrogen ions and some oxygen. Okay? So, when we're looking at the light-dependent reaction, we got a couple things going on. So, in, we have H2O. And then out, we are having O2. Now, the hydrogen ions are going to be used to generate an ATP. So you've got this little machine called an ATP synthase that is going to use these hydrogen ions, and then what you're going to end up with is you are going to make an ATP. Now this electron, on the other hand, what's going to happen is that it is going to get transferred from molecule to molecule. And so it's going to go here to here to here, and eventually it is going to get made into something that we call an electron carrier. And in this instance, that electron carrier is NADPH. Now, an electron carrier, all that means is that it is a molecule that has an electron in it that it does not want to keep. And so what happens is that it's just full of energy and it's going around all over the place, and it just can't wait to get rid of that extra electron into something else. And so these electron carriers do exactly what their name says. They carry an electron to another reaction where it can be used. So the purpose of this whole entire first thing is, even though we use our water and we use our oxygen, the main purpose of all of this is so that we create these two things, the ATP and the NADPH. That is the purpose of the light reaction. All of this other stuff, all it's really doing is that it is going to be for the sole purpose of making ATP and NADPH. Now, once we've created that, the ATP and the NADPH are going to go into here, which we call the stroma. Okay, and the stroma is just a type of cytosol inside of there that holds different kinds of things dissolved inside of it. All right, so now we're going to go into the stroma with our ATP and NADPH, and we are going to move over to the next reaction, which is called the Calvin cycle. Now, this one is kind of confusing. Well, I don't think it's confusing, but a lot of times when we have things named after scientists, they're not self-explanatory. And so you're just going to have to memorize that the Calvin cycle has to do with photosynthesis. Okay, so the next thing that you, with the Calvin cycle, again, it's going to happen in the stroma, is that you are going to bring in your CO2, and then you're going to take your ATP and your NADPH that you got, and you are going to put it in a cycle. Okay? Now, there's some leftover starting stuff over here that has another carbon molecule that comes in, but it doesn't have enough carbons on it to be glucose. And so what happens is that your CO2 comes in here, your ATP uses its energy, your NADPH uses its electrons, and by the end of this cycle, what you end up getting out of it is that you end up getting C6H12O6. I'm not going to go in all the steps. You don't need to know all the steps. All you really need to know is that you lost your ATP and your NADPH in this cycle, and your out is going to be C6H12O6. So, Calvin cycle should be fairly quick for you. You just need to realize that you are using your carbon, okay, using the carbon for photosynthesis, which is carbon dioxide, and then you're getting a new carbon molecule out that has a lot more stored energy in it called C6H12O6 or glucose. All right, now we want to be able to get into cellular respiration. And cellular respiration is a little more complicated. There's a few different things that you guys need to be able to understand in here, and it might get a little tricky for you. So let's just go through this real fast, and I'm going to give you a little bit of a flow chart for this of where we're going to start out with cell respiration. Okay, and we are always going to start with a step called glycolysis. Okay, now this glycolysis, a couple different things, is that you need to look at the name. Glyco means glucose. Lysis means to split. So this word means to split sugar. So what are we going to do in this first step? We're going to split sugar. All right, now, here is where things get a little tricky. It is because glycolysis is going to take place in the cytoplasm outside of the mitochondrion. And then, basically, to get into the mitochondrion and use the processes in there, you're going to need oxygen, okay? So if you have oxygen present, then you'll be able to go into the next step, which is called the citric acid cycle. Okay? Sometimes I just refer to it as the CAC. All right, so glycolysis, if you don't have oxygen, then you're kind of in a hurting spot of where you don't have enough stuff to keep getting energy and stuff. And so then we move to something called fermentation. Okay? And now with fermentation, that is where you are going to just recycle some of the products from glycolysis so that you can keep on splitting sugars. And when we go through it, it will make a little bit more sense, but I just want to get this flow chart out of the way. And there will be two different types of fermentation that you will need to know about. Number one, you will need to know about alcohol fermentation, okay? And really all that's saying is that the end product is going to be alcohol. And then the other one is lactic acid fermentation. And again, the product of that is going to be lactic acid. Okay, so if you do have the O2, you do your citric acid cycle, eventually the last thing that you're going to have is that you are going to do the electron transport chain. And the electron transport chain is probably the most important step of all of these, just because it makes so much freaking ATP. So just remember that. Everything else is building up to that electron transport chain, and it is very, very useful and very productive when it comes to making energy. So, let's start into the individual steps now. All right, so here we're going to draw our mitochondrion. Okay, and then what's going to happen is that our first step is going to be glycolysis. And glycolysis is, again, as we talked about, is going to mean splitting sugar. Okay, it's going to split that glucose in half. Now, to get that started, it has to spend ATP. However, by the end of it, you're going to make more ATP. So when we're talking about glycolysis is going to happen outside of the mitochondria. So it's going to happen outside of the organelle in the cytoplasm. Okay? And what we're going to end up with is a couple of different things. So number one in this, our in, is going to be our C6H12O6. Okay? And our out, we are going to end up with 2 ATP, and we are also going to end up with some electron carriers. So we're going to end up with two NADHs, and NADH is the one that's used primarily when we're talking about cellular respiration. Okay, and the other thing that you're going to get out is that you are going to get out two pyruvates. Okay, now that might be confusing. It's a word that you've never heard before, but really all I want you to know is that it is a 3-carbon molecule. So we went from a 6-carbon molecule here, and now we have two 3-carbon molecules here. Okay, and we ended up with a little bit of this and some of these. Okay, so where I want to go with from this is if we don't have any O2. Okay, if we don't have any O2, what is going to happen is that now we're going to stay out inside of the cytoplasm, and we are going to have to do fermentation. And fermentation, all that fermentation is really going to do is that it is going to do glycolysis. Okay, so we've talked about glycolysis. So it's going to do glycolysis, and then what it's going to do is that it's going to go down, and it's going to turn your pyruvate into either lactic acid or alcohol. Now, what is going to happen as you're changing that into lactic acid or alcohol is that your NADHs that you made are going to get recycled into their starting material, NAD+, and then it can go back into glycolysis and keep on breaking apart stuff. So that is really important, because really all fermentation is recycling stuff so that you can keep on making energy in the short term. You run out of energy, what's going to happen? Well, you're going to die. Okay, so you want to get even though the 2 ATP isn't a whole lot of ATP, 2 ATP is better than 0 ATP. So you'll keep on doing that. Now, the only other thing that you need to know from this is perhaps it might show up, it might not, but when you have alcohol, alcohol is actually a 2-carbon molecule. So where does that carbon always come out as? Well, right here, as it's being changed, you are going to lose a CO2. Okay? So that's fermentation. I don't really want to go into it a whole lot, other than you just realize that you have to keep on doing glycolysis if you don't have oxygen, because that's the only way you're going to be getting energy. If we have oxygen, then we go inside of the mitochondria. Okay, so here we have our mitochondria, and what's going to happen is that the citric acid cycle is going to happen inside of the mitochondria. So we've got the citric, too close to the top of the screen there, acid cycle, okay, and then during the citric acid cycle, what's going to happen is that you're going to take your pyruvates and you are going to switch them around a little bit, and then, so you're going to start with your pyruvate, and then you're going to go to like this 6-carbon, which is citric acid. Okay, and then what's going to happen is that you're just going to go through a cycle of where you break off a CO2, and then it becomes a 5-carbon molecule, and then you break off another one, CO2 comes out, and now you have a 4-carbon, and it stays like that until you go back up. Okay? So, the key part to the citric acid cycle, though, is that even though you're getting out CO2, so our out in this case, in the citric acid cycle is CO2. The thing about this is that every time you're breaking off one of these CO2s, you are going to be getting either electron carriers or electron carriers and ATP. So by the end of this whole process with the citric acid cycle, you are going to end up with 8 NADHs, and you're going to end up with 2 FADH2s. Okay, and that is just another type of electron carrier. And you're also going to end up with 2 ATP. And so this cycle, again, we get some more ATP, but the key part of it is that we are making lots and lots of electron carriers. Because that will bring us to the next part. Let's see, I guess maybe we'll just start a new page. The electron transport chain. So we started out with 2 NADHs from glycolysis, and then we had 8 NADHs from the citric acid cycle, and then we had 2 FADH2s from the citric acid cycle, and that's it. Okay? So, the electron transport chain, it's going to look very, very similar to the light reaction of photosynthesis. It's just going to be a little bit different. So, you got your mitochondria, and then you've got this part of it that's just a bunch of membranes. And so, the ETC is going to happen in those membranes. Okay? So, during the ETC, what happens is that all of these electron carriers are going to lose their electrons, and they're going to get put through and make a whole bunch of hydrogen ions, and then the electrons are just going to take all of those hydrogen ions and make a concentration gradient. And then as that concentration gradient gets fixed, then a couple things are going to happen. So, number one, you're going to get out H2O, because those hydrogen ions that you get from here, okay, these hydrogen ions have to leave somehow, and they leave in the form of H2O. Now, what happens is that as it goes through this process in the electron transport chain, is that you are going to get, for every NADH, you're going to get 3 ATP. And then for every FADH2, you're going to get 2 ATP. So if we figure that out, we have 10 of the NADHs. That's going to equal 30 ATP. And then with our FADH2s, we have 2 of those. So we get 2, I'll fix that, 2 times 2. And so that's going to equal 4 ATP. So just from the electron transport chain, we are going to get 34 ATP. Now, if we take that, we've got our 34 ATP from here, plus 2 ATP from glycolysis. And we end up with another 2 ATP from the citric acid cycle. That, for the whole entire cellular respiration, we're going to end up with 38 ATP. So, this is definitely the best way to be able to make lots of energy and be able to supply the energy necessary for us to survive.